Good morning, everyone. Thank you for participating in today's webinar from Drs. Evie Mattei, Stephen Moore, and Ella Temprosa, which will focus on COMETS Analytics, an introduction to the analytic platform from Metabolomics. Before introducing our speakers, I want to quickly review a few logistics. Should you experience any technical difficulties, you can contact us through the questions box in the expanded control panel, or by phone or email. If you need to view live closed captioning, please click on the link that will appear in the chat box. At the end of the presentation, we will allow time for questions. If at any time during the presentation you have a question, please type it into the question box and I will ask it on your behalf during the Q&A session. So I plan to keep today's introductions relatively brief because we have 3 speakers, all of which have been very involved in COMETS. So most of the group should be familiar with them. Having said that, it is my pleasure to introduce Drs. Evie Mattei from The Ohio State University, Stephen Moore from National Cancer Institute, and Ella Temprosa from George Washington University. Dr. Temprosa will be our first speaker, followed by Dr. Moore, then Dr. Mattei. So we'd like to welcome the 3 of you and all of the attendees to today's webinar. I'd like to hand over the presentation to Dr. Temprosa.
I'm sorry, I was on mute. Thank you, Krista, for the nice introduction, and thank you all for your interest in COMETS Analytics, a reproducible and efficient platform for conducting consortium-based metabolomics research. Can you guys hear me?
Yes, we can hear you.
Okay, thank you. So our presentation today is in 3 parts. I will address the why of COMETS Analytics, then Steve will show you how COMETS Analytics work through a demo, and Evie will conclude with what the magic is under the hood. So in the next few minutes, I will describe to you the principles that motivated the design and the infrastructure, as well as a sample workflow that each analysis cycle will go through. And at the end of the presentation, we hope to convince you that this new paradigm in conducting consortium research has great potential to meet COMETS' goals to leverage its resources to accelerate scientific discovery. Since its inception in 2014, our consortium has grown to include 41 cohorts. A major goal of COMETS is to produce jointly coordinated, multi-cohort, high-impact publications describing relationships between metabolites and diseases in diabetes, cardiovascular disease, and cancer, and other biological constructs such as aging and genetics. Many of you have participated in various consortiums where patient-level data are sent centrally. Challenges in these models include the dreaded data use agreements, central data harmonization by folks who don't have intimate knowledge of the data collected, possible data stagnation over time where it's a one-shot deal that the data is being sent in and it might not be updated over time, and the main problem in the harmonization of metabolite levels across platforms and samples. At the outset, the COMETS Steering Committee decided on no patient-level data sharing, so we had to find another model. The COMETS Data Harmonization Group, which I chair, more recently with Evie, was given the challenge for figuring out how to coordinate the data-related activities of COMETS. So our challenge was to provide an infrastructure to facilitate COMETS goals and coordinate data analysis efforts. So our main big vision was that maybe a disseminated, cost-efficient platform can help, meet this challenge. So here we have COMETSanalytics.org. It's been in testing mode for the last few months, so we thank the cohorts that have been testing it and really making it a better platform for the rest of the consortium to use. So although the URL was chosen by necessity, comets-analytics.org, since other websites were already taken, it has a nice ring to it in the age of big data and data science. So here are the guiding principles for the infrastructure and design for COMETS Analytics. In data autonomy, the most important thing is the data stays within the cohort, but we have centralized code to conduct the analysis, and we have user registration for the security and monitoring of activities. We wanted COMETS Analytics to be simple, useful, and efficient. We employed key user experience and user interface design principles to make COMETS Analytics simple and easy to understand. We used interactive control for useful exploration of analysis results, and efficient, obviously, because we want to have minimal burden on current cohort resources. Because in the way that it's set up, we can run through multiple analysis for multiple projects at the same time. COMETS Analytics was also designed to be scalable and sustainable. The application lives in the cloud, where we can monitor usage and load and adjust resources to meet longer-term needs. The analysis we will conduct in COMETS Analytics support reproducibility by using common code and metadata, which all reside on GitHub, so everybody can see what analysis and the approaches that were done. Furthermore, the templates are available for others, so once a paper is published, the analysis template is available for the public to use to perhaps conduct the same analysis for their own validation of their own data. So here's what the toolset looks like. It's a cloud-based application through Amazon Web Services. They have an R server on the cloud. We use RStudio Shiny for the prototype. And the main analysis functionality is facilitated through an R package in COMETS, which we will make public shortly. The front end, which the user interface was developed by NCI CBIT using HTML5, and again, the GitHub houses all the code as a code repository. So here are the COMETS Analytics cycle and features. We start off with Metabolite Harmonization, where we create mapping of metabolites across cohorts and platforms. Data prep, which is on the end of the cohorts, where we facilitate creation of common input files to go into COMETS Analytics. We check for data integrity at the time of the analysis to identify possible data problems prior to running the models that we need for each project. Then, once everything checks out, then we conduct a cohort-specific analysis, which uses patient-level data that you submit through the website, but it's not kept on the website. So here we have also facility for data exploration and will be used for approved manuscript or project proposals. And at the end, you will get this standard output that we will use for meta-analysis. So these, you will just have to send us the results centrally, and Steve and IMS will be in charge of the aggregated cohort data, not the patient-level data. So in metabolite harmonization, so the first stage of this cycle, as I said, is to maintain an up-to-date master list of metabolites, which is primarily led by IMS. So now it's Nathan Appel who's facilitating this work. And in this process, we are able to have a master metabolite list, which includes important information like HMDB, KEGG, and other metabolite information. We also are able to pull in information from the Metabolomics workbench to classify the types of metabolites, and as well as Metabolon if that data is available for those with Metabolon platform. In the harmonization, we've been proud to to successively increase the number of metabolites that are harmonized. So at present, we have about 4,000 metabolites harmonized, And in recent runs, we're finding that greater than 90% of metabolites are harmonized. So that's a good thing for less work during the meta-analysis stage. So here's the platform that we have according to the identification. So these are only the identified metabolites. We have about 1,500 from Metabolon, 835 from BrainShake, and Broad 500, and so forth. And if you look, there are metabolites that are, for example, Metabolon and Broad have common metabolites of 121, with Biocrates 24, collocated WCMC platform 92 is common with Metabolon. This is, of course, increasing as we expand the harmonization list. What we found is that lipids primarily drive a lot of the metabolites. There are quite a few phenobiotics and some amino acid peptides. This is the distribution of the top 8 metabolite classes. So about 40% are lipids. In terms of data preparation, this is where each of your cohort will have to assemble their data to a standard Excel format so that it can be processed in COMETS Analytics. We multiple data inputs are available, like Excel, Data, SAS, or R. They can be used. We also have this Create Input tool that allows you to use your source data file to compile this Excel format. So Evie and Steve will go through this quickly, but just to show you, we have a standard format spreadsheet that has 5 tabs, and we have a Create Input here where you can create your own Excel file to be put into COMETS Analytics. In terms of data integrity, this is at the stage where you submit the data through COMETS Analytics, and we want to validate the input file before analysis to make sure that there's correct variable mapping, that there's correct metadata on metabolites, to make sure model specification was done correctly, so that, for example, an adjustment variable will not be the outcome specified in the models. And it gives you warning and meaningful errors for you to address those issues. And Evie will talk about this more in detail. Then the magic happens. This is where the cohort analysis happens, where we have 3 nodes. You can specify your models interactively, where you can specify, what variables you want to look at using point and click, and batch mode, where you can use the pre-specified models in the Excel sheets to look at what the results are, or the super batch, which is the one that we'll be using for most analysis, where you submit the job to a server and you will get the results in your email. There's some other cute features in the analysis where you can tag certain metabolites to look at them more carefully. We employ exploration controls so that you can do interactive analysis of your results. And there are modules for correlation with heat maps, hierarchical clustering, and linear models. Here's kind of what it looks like in interactive mode, but Steve will go through this quickly on his presentation. This is what it looks like, but Steve will go through it. Here's a correlation heat map under interactive mode, and you can play around with this, zoom it, look at certain filters, that kind of thing. In terms of meta-analysis stage, this is where we synthesize the aggregated cohort results. So after you run your analysis, you will send it centrally. We have a standard output format for each cohort, and this will help facilitate the meta-analysis more easily. In this analysis, we will consider random or fixed effect cohort effects. And after publication, where we will, publish the results online of the cohort-specific results, the public are then able to run the same analysis on their data for validation. So that's the full cycle of the COMETS analytics. Now we're looking forward to the 2020 COMETS strategic goals, which was assigned to the data harmonization group with 2 goals. The first goal is to characterize exposures and metabolomics, and this objective came from the questions of how exposure relates to metabolism as assessed by the metabolomics approach. And the fourth goal in that strategic goal is to identify COMETS' current resources. So as we go through different projects and go through the cycle, we are envisioning that we can achieve this goal through this tools or projects that we're about to embark on. So one is to develop a database of accumulating results on the meta-analytic and cohort-specific levels, and we hope to make this public once the paper is published. Goal 4 will help identify COMETS resources, and in this project, we hope to develop searchable databases for inventory of resources, including what cohorts are in COMETS, metabolite and data availability, which is facilitated by the harmonization file that you sent to us, and the data tables that Bing, you, and Steve had compiled for the design paper, which we all saw, the different projects that have been approved, abstracts from COMETS, publications from COMETS, as well as the sample availability that Krista is heading up. So in summary, COMETS Analytics provides a new paradigm in conducting consortium-based research that we hope promotes reproducibility, scalability, sustainability, and validity to advance our understanding of the metabolomic basis of diseases. With that, keep in touch and let us know what you think. Thank you. Steve will then now walk you through the software and explain the different aspects of COMETS Analytics.
All right, thank you. Can you hear me first?
Yep.
Okay, fantastic. So thank you, Ella, for the great introduction. I'm really excited to present COMETS Analytics to all of you. I have just one slide to start, very short. As Ella mentioned, we're going to be doing the analyses within COMETS on a disseminated basis. This approach can be challenging, though, especially if the underlying data are complex themselves, like they are for metabolomics. So COMETS Analytics is designed to facilitate disseminated analyses in 3 specific ways. First, COMETS has built-in checks and nudges to help investigators provide high-quality data, so enhance quality control. Second, it's simple to use. It doesn't require learning a programming language, and this helps ensure a broader participation. And third, the data are output in a meta-analysis ready format, which should help to accelerate the analyses. So as I walk you through the demo, I hope that you'll be able to see these features in action, and I'll call out a few as I go. So without further ado, the demo. All right, so this is the login screen. It's at cometsanalytics.org. You can log in using either a Facebook or a Google login. You can also register for a new ID if you don't want to use either of those. I'm just going to use my Google login. Oh, sorry about that. Let me just, there we go. All right, this is the welcome screen. It provides a brief introduction to COMETS Analytics. We're keeping it short, but in the future, this is going to include some other explanatory materials, including a tutorial and possibly a video archive of this presentation, maybe a frequently asked questions section. So currently, COMETS Analytics has one primary function, which is to produce correlational analyses, and that's at this tab here. Later this year, we're going to add logistic regression and some other functions, but for now, it's just the correlations. To help get people familiar with COMETS Analytics, and this is very important, we've included a sample metabolomics file that's already ready for analysis. And so that's what I'm going to use today for the demo. Go ahead and download that. The file is simulated based on a real and relatively large data set. It has 1,000 participants and 600 metabolites. I'm going to open it. We prepared this file so that users can test drive COMETS Analytics, get familiar with it, run some analyses, and do all that without having to create a data file of their own. So it gives them a way to get familiar without having to incur a high startup cost, basically. So I'll provide a quick tour of some of the inputs now. Evie is going to go into a little bit more detail on the last two, so I'll focus on the first three. So the first tab is the Metabolites tab, and it provides the metadata for the metabolites. So this first column, I'm going to page up here, is just the ID of that metabolite in our data set, and then these other columns include metadata about these metabolites. So this would be the actual name, for instance, the pathway that it contributes to, the subpathway. Most of this information is not mandatory. In fact, I think the only thing that is mandatory is the ID, but we're trying to collect as much information as possible about these metabolites so that we can facilitate harmonization over the meta-analysis. And in particular, of value are these IDs, comp ID and HMDB ID, which makes it possible to link metabolites where maybe the names were changed slightly. There's some metabolites that are called different names depending on what continent you're in. Having these IDs allows us to make a linkage. The subject metabolites are just the metabolite levels for the different subjects. So subjects are rows, metabolites are columns here. And then the participant covariate data is in this table here. So that's the first three tabs. These are just very simple things like age and gender and smoking and so on. So those are the three tabs that are essentially mandatory. Without that information, the analyses won't run. But we also have two other tabs that help to coordinate meta-analyses. So that's the var map and the models tab. So the var map tab isn't strictly necessary, but it essentially contains the whole map for how the metabolites are coded in the data analysis that you're doing. This tab and the model tab are typically set up by the PI in advance. So the PI would set up these tables, send them to the analyst at the different institutions, and they just slap it into this Excel spreadsheet. They don't have to make any changes. And it tells that analyst or user how to code the different variables. So for instance, female, it's specifying that they want a code of 0 for male, 1 for female. For smoking status, a 0 is never smoker, 1 is former smoker, and so forth. So Evie will go over these in more detail, but that is the basic gist of that. And the specific reason that it's set up this way is so that the PI can set up all of the analysis in advance. Let me go through the models tab, sorry. All of the models and all of the coding in advance, which helps ensure a higher level of standardization and data quality. So in the models tab, just as an example, these are all of the models that will be run for a particular analysis, and that analysis is of age as it relates to all metabolites. All metabolites are the outcome, age is the exposure, and then these models are adjusted for different factors here. And they also can include some stratifications as well as some where statements. All right, I'm going to move back to COMETS Analytics now. So for the demo, actually, I need to make one quick modification to this spreadsheet just to make sure that it runs all the way through in time. Okay. So you have to specify a cohort. I'm selecting the file that I just downloaded and just modified ever so slightly. I'm now performing an integrity check. This takes just a few seconds to run, but it provides basic checks ensuring that the metabolite names match across the different tabs and that the covariate names match across the different tabs. So once this integrity check is complete, the user can then move on to an analysis. And in that spreadsheet, actually, one let me take a step back here. So if the input tables are set up correctly, the analysis that we want the consortium users to do is very simple in a blink and you'll miss it kind of way. And what we want the user to do is, first make sure that this batch mode is clicked. Then from this choose model, select all models, put in user email address, and click run model. And when the user does this, it sends the job to an Amazon server and a queuing system. It runs all of the models, and it sends a zip file to my email. So assuming that everything is working well, and we'll find out in a few minutes, we will come check that zip file out shortly. But in the meantime, behind the scenes, there's actually a pretty substantial amount of analysis that's going on, and I can show you what's happening through our interactive mode. So the first model in that spreadsheet, so we're on interactive mode here. I'm just going to change this to gender adjusted. That's our description. There's nothing special about that. It's just a label. The first analysis was of age and all metabolites. So I'm just going to replicate the same thing here and adjust it for gender. And I'm going to click down here, run model. So this model is exactly equivalent to what was in the spreadsheet here, gender adjusted model, outcome of all metabolites, age as the exposure, adjusted for gender, no stratification, and no other where statement information. And it's running through all 611 of these metabolites and producing an age and metabolite correlation. And so this will finish up in just a second. Now, back at this website, the analysis is showing us the correlations and p-values. And I'm going to sort by the p-value. And on the top, we have this metabolite, vanilla mandolate, or VMA. It's got a correlation of 0.323 and a p-value that's highly significant. And there's a number of statistically significant associations you can see. And a little bit lower down, we have C-glycosyltryptophan, which is a metabolite that's been identified in prior publications as associated with age. And if we scroll down just a bit further, you can see that there's many different pages reflecting the fact that there's 611 different metabolites. So alternatively, if you want to see everything at once, you can download the results as a single CSV file. All right, so that's here, and we sort by the p-values. You've got the same thing at the top, VMA, correlation of 0.323. All right, so that is the kind of core functionality of the interactive mode, but there is one other feature which I wanted to highlight really quickly. So let's say that you want to do a little bit of additional exploration. You can do that graphically through the heat maps that Ella mentioned. So I'm just going to plunk in a maximum p-value of 5 zeros and a 1. That limits us to the top 38 of the metabolites. I'm going to tag them, and we'll just call this age metabs. Okay, so we now have a list of the top age metabolites, and you can then run a correlation of those metabolites on themselves, or anything else for that matter. So I'm just going to do that quickly. It takes about 10 seconds to run, so hang tight. All right. So I've fed these metabolites back into the model. We can change to the heat map tab. And I like to do this with hierarchical clustering because it gives it a little bit more elegance. We'll switch this height to about 800, the width to about 1,000. All right, and we can now, I'm going to go a little bit wider even. All right, and we can now explore how these age-related metabolites are related to one another and look for potential clusters. So a few things kind of immediately strike the eye. First, there's a little cluster right here. So we have this methylglucopyranizide, ceiloinositol, stashedrin, these are all citrus metabolites. So it turns out in this particular population, older people consume more citrus for whatever reason, and you can easily see that just in this cluster of metabolites that pops up. There's another cluster here, which includes erythritol and threatol. I actually think that this may be dietary as well, based on some of their analyses that I've done. And then finally, the biggest cluster is down here, anchored by pseudouridine. This is an inflammatory marker. Also part of this cluster is C-glycosyltryptophan. You can see how these metabolites, these pairwise correlations, relate to one another just by looking at these pop-ups as you scroll over. So that's our heat map functionality. Allows you to do a little bit more exploration than maybe the app would be able to do just through the batch mode. All right, so speaking of that, I'm going to shift our focus back again to the left-hand panel. And we've been doing interactive mode analyses. Let's switch to batch mode. Now, before I ran the all models, but underneath this are actually each of the individual models as well. So I can select gender adjusted, for instance, and run just that. If you select anything other than the all models, it will actually run it right on the screen for you in real time. So this will take probably about 15 seconds more. Hold on. All right. So by running the gender-adjusted model, what it's doing is it's pulling it straight off of the spreadsheet. And again, just to go back to the input, it's pulling it right from here, gender-adjusted, all metabolites, age female. And it's running the exact same thing that we just did in the interactive mode. If I sort by p-values, again, we have VMA on the top, correlation of 0.323, same thing as before. All right, so this is just an alternative way of running the models that doesn't involve point and click, but actually pulls it directly from the written text on that spreadsheet. Okay, so circling back to that spreadsheet, if, for instance, I run the gender-adjusted model on that pull-down, it's going to run all metabolites, age and female. If I run the multivariable adjusted, it's going to do the same thing, except with these adjustments instead, and so forth. It'll just follow the instructions that are provided in this spreadsheet. Now, circling back to the very start, we had selected all models, and I said that it would produce a zip file, and it's going to send it to my email. This particular analysis takes about 5 minutes to run, and as long as nothing is in the queue, it should be right here on top. All right, fabulous. So this is the email, just so you get a good look at it. It ran through this COMETS input 51. It sends a link to a zip file, and it includes just a few messages. It takes 300 seconds to run this particular set of models. It also includes these warnings, which are not really warnings. They're more like information about your data set. So recall that in the models, they're all set up by the investigator in advance, and it's meant to apply to, say, a set of 46 cohorts, right? So for some cohorts, you will have adjustments that aren't strictly necessary, and that was the case here. So we have a gender-adjusted model, but this data set only has women. And so what this warning, in quotes here, is essentially telling you is that you only have females in your data set, so it's not going to adjust for gender in the models. That's all. All right, so let's pull up the zip file. All right, so the zip file includes all of the different models on that original spreadsheet, nicely packaged and bundled. Just to bring the point home, if I sort by p-value again, I will get the same thing on top, VMA, correlation of 0.323. It's the exact same thing, except that now it's bundled in a zip file with all these other results. If we look at, say, a stratified model, you will see that essentially it's run the correlations within a smaller subgroup. That smaller subgroup is BMI group level 1, which is the lowest BMI group. And if you go all the way down, you'll see that after about 611 metabolites, you're on to the next BMI group. So this is essentially all of the metabolites run against age within strata of BMI. And there's similar findings in all of these other different models. So one more thing, and then we are done the demo. 2 additional details, actually. So we are working on a new version, and this is what the output would look like from this new version of COMETS Analytics. We've included not just an analysis of age, but also a body mass index. One of the features of COMETS Analytics is that it's very easy to just add more analyses to each of the spreadsheets. And we have additional descriptive information that's supplied to the PI, basically, the person who's doing the meta-analysis. So one of the issues that a meta-analysis like this might run into is that you don't have very much descriptive data about each of the individual cohorts itself. We took the opportunity to kind of run those analyses so that we can provide that to the PI. So for instance, in this particular data set, I mentioned that it was only females, and so we have 1,000 people, and it's 100% of the cohort. It provides similar information about, in this case, never smokers. There's 576 of them, constituting 57.6% of the cohort, and so forth for the other covariates. Also produces some basic univariate statistics for some of the other variables like age. And there was one other thing as well, which is that we now provide to the PI all of the inputs minus the subject metabolites data. So I mentioned how the COMETS Analytics system includes a number of checks and nudges to make sure that the investigators are doing things correctly. And this is kind of one of those checks. By providing all of the metabolites to the PI who's doing the centralized meta-analysis, all of the VAR map, and all of the models, the PI can actually check the models and make sure that the correct thing was being run. So with that, I've come to the end, and I think I can turn it over to Evie.
Can you hear me?
Yes, I can hear you.
Okay, great. All right, so hopefully now everybody has a clearer idea about how to use COMETS Analytics, and I want to take some time and talk a little bit more about what goes on behind the scenes, particularly talking a little bit more about what goes into the software, what comes out. And a lot of the software was really designed with reproducibility, usability, and the consistency of the input and output. And in terms of the consortium, that's very important when thinking about doing the meta-analysis and ensuring that all the data is formatted properly going in and that the output is consistent for performing the meta-analysis. So I'd like to go back a little bit to the data input. So you've seen the Excel spreadsheet, and there are 5 sheets. So 1 being the metabolite meta information, 2 being the subject metabolites information, 2 being the subject metabolites information, right, with the metabolite abundance data, 3 being the subject meta information, 4 being the VAR map, and finally 5 being the models. And so I want to focus more about this VAR map sheet here, which I think is very important for doing the analyses in the consortium. So this is a close-up of the VAR map sheet for the example input file that we provide through the COMETS Analytics software. And there are 5 columns to this sheet. So the first column is called VAR reference. And this is something that is predefined for you by basically those that are organizing the meta-analysis. And these are the variable names that are to be used across the cohorts. So as a user, you don't need to change this VAR reference name. And if there are multiple cohorts that are doing the analysis, For example, the cohort subject ID is always going to be called ID, and the metabolite identifier is always going to be metabolite_ID, which is shown here. And so these are predefined for you. The definition, of course, is also predefined. So this is where the information about the coding is there. So for example, for females, we have 0 are coded as males, 1 are coded as females, and then you see the other definitions here, again, that are typically predefined. So next, the next 2 columns are really about for the user. So here, the cohort variable, this is the actual column from your data. So if you look at the first row here, for the cohort subject ID, you don't have to call it ID in your data. Here, for example, in this particular input, the column is called sample ID in, say, my own data. But I want to be able to map the ID that I used to the ID that is used across the cohort. The var type is also important because all it says is whether the variables are continuous or categorical. And this ends up being required because all of this on the back end is written in R, and R will treat categorical variables and continuous variables very differently in terms of the linear model. So even if you have numbers, for example, here, female, for gender, you have numbers 0 and 1, but you want to explicitly tell R that it's categorical because you want to treat it as categorical. And the way we can check that is to check this var type column here and make sure that the analysis is done correctly using the proper variable type for that particular variable. And then finally, there's additional notes, and these change again by ID. These are notes that you could add yourself. These are notes that could be added a priori, but it could have information, for example, about missing values or what to code the missing values as. So next, the model sheet is where you define the model. So Steve has already talked a little bit about this. The nice thing with this is that it's very flexible, and you can add to it if you want to run some analyses independent of COMETS, and so the first column here just has a name for the model. The second column has the outcomes, then exposure, then all your adjustment variables. So here, note that the adjustment variables are separated by a space. And then here you have the stratification variables. And if you can also have multiple stratifications here, and they would need to be separated by a space. And the final column here is where. So if you note row 10 here, which is model 2.7 HT stratified. And so here, we're only wanting to look at the females group. So we specify where female equals 1. And you can also specify multiple where arguments here as well. So note that all the variables that are in these models, they are the variables from the var reference in the var map sheet. So let me go back. They are the names here. So they're consistent. So this model sheet, once it's set, then it can be used across many different cohorts. And then the cohorts, all they need to do is map their own IDs to this var reference ID. So there's an example, a sample data input, as Steve already pointed out, but here if you're at the front page, you know, it's below here, just underneath the help. And of course, you know, when putting this sheet together, there are some constraints that need to be thought of. And first, if you have your subject metabolite, which is the metabolite abundance sheet, and you have the meta information for the samples, the subject IDs that are used in the sample names and in the abundance sheet need to match. And same thing for the metabolites. So the IDs that are used for the metabolite abundances should be the same as the IDs used for the meta information. And these requirements are just so it's possible to merge the two and to link the two. And finally, I've stressed this already, but in the VARMAP sheet, all the IDs that the user puts in, those IDs need to be present in your data, in the other sheets. So in the sample metadata, in the metabolite abundance data, and also in the metabolite metadata. So this probably sounds a little bit complicated and overwhelming, and realizing this, we built this utility that helps you create the input. And this utility here, so this again is the front page, and if you look at the bar up here on the right, there's this create input utility. So if you click on that, you'll see this, this window that opens up here, and it's requiring you to input 3 files. And these files all need to be in CSV format, comma separated values format. And the first file that you put in is the metabolite metadata. The second file you put in is the metabolite abundances. And the third file you put in is the subject metadata. And then you can select a template. So if you want to do analysis for the age, then you would click this. And what this does is it automatically pre-fills all the model sheets, so all the models that should be run for the age analysis. And it also fills in the VARMAP, those first 2 columns of the VARMAP that are pre-designed for you. So basically here, if I select age, then there's more information that you need to put in. Okay, so here I've selected the template age. And then it asks you to select the ID. It selects you to select the metabolite ID, then the variable for the age, the variable for the female, and it goes down. But for every time it reads in your data, it takes in the column names, and so it populates your options. So in this case here, for the smoking group, you would go down and select what you have called the smoking group in your data. And in this case, we called it the same, but you may have called it just smoking. So you would select smoking down here from the pull down. And it goes through every single variable that you should have for the age analysis, because that variable is going to be used in the model. So this utility has a lot of checks and balances that are built in, and it just helps you do those mappings. So if you use those utilities, you can directly use the output from that and then input that into your analyses. Okay, so now that you have your input and you are starting to run your analyses, there's a series of integrity checks that go on, and that's all behind the scenes. And we've put in quite a bit of effort to try to return meaningful errors when there are. And so some errors that can occur is, for example, incorrect mapping from your VARMAP sheet. So say, for example, you map an ID that is not present in your data, it's going to throw an error. If the IDs from your metabolite metadata and from your metabolite abundance data do not match, it's going to throw an error. If the models do not make sense, for example, if you try to incorporate an adjustment variable that is the same as the outcome variable, that's going to give you an error because you cannot do that. So there's a lot of error checking that goes on behind the scenes, and if there is an error, this here, for example, is an error, and this is what it would look like. So here I'm just going to read it. Here the error is that the user input ID in the cohort variable column of the VARMAP sheet is not found in the subject data sheet. So check your input file. So you would go back to your input data in the VARMAP sheet and check this cohort variable for the input ID, and then change it accordingly. So Of course, if you do get a non-meaningful error, please let us know, because you know, we've run a lot of checks, we've run a lot of different types of data, and the only way this software gets better is through user input. So if you see something and you have no idea what it means, let us know, and hopefully we can incorporate it into the integrity check so that to make sure that we return meaningful errors. So the harmonization, just, you know, both Ella and Steve have touched on this, but again, you know, this goes on behind the scenes, and it checks that your metabolites ID against all of those that have been harmonized in the group. And I do want to note that even though your metabolites may not have been found previously in COMETS or hasn't been mapped, the analysis will proceed with those regardless. So it's not a requirement to have the metabolites harmonized, but the harmonization does happen automatically. And it will output the results of the harmonization. So for example here, there were 350 metabolites that were input for the analysis, 117 were harmonized, 233 were not harmonized. This is an old example, by the way. The total harmonized now has, you know, skyrocketed compared to this example. But then there's the option to download the list of metabolites and send to the harmonization group. And a lot of folks have already sent us this, and this is what helps improve the harmonization master file. And I would encourage everybody to, you know, to keep doing that so that we can get this harmonization as close to 100% as we can. So the current status, again, this is the link for COMETS Analytics, and we have the first version that is deployed and completed. It's version 1.3. And CBIT is building and supporting the web interface. And so they're providing secure access. They're allowing multiple users to use the software at once. And a lot of the nice GUI interface is supported by them as well. The next version that we're working on right now will include some new features. One, it will allow you to do batch processing on the command line. So right now it's possible on the GUI, but if you do not want to use the web interface, you could do that as well. So this is, again, more of a behind-the-scenes task here. Second, we are working toward incorporating linear modeling and logistic regression in addition to correlation analyses. And then third, we will be incorporating pathway analysis. So this becomes important as we're trying to interpret the metabolites that are significantly associated with the outcomes of interest. So I do want to point out that there is this nice GUI interface for those that do know R and do not want to use the GUI. All the code, vignettes, and detailed documentation are on GitHub, and it will soon be released, while we submit the paper or even perhaps before, and it's on GitHub. And so you could, it's basically a two-liner install. Sorry, you have to download this package called DevTools, and then you can just install it directly. But all the functions that are used by the GUI interface at the back end, it's all going to be in the R package. So in terms of reproducibility, in terms of transparency, and the best practices for doing, biomedical research and these large epi research, this is quite key. So with that, I would like to thank you. There's lots of people that are involved in this effort, and I want to give a special thanks as well to the users, and keep giving us feedback, because this is how we're making the software better, and so we certainly appreciate it. Thank you.
Thank you so much, all three of you. We do have a number of questions. We have about 3 minutes, but I will ask, so far we've gotten approximately 5 questions. I will ask those, and then we may go over a little bit. Obviously, if people have to leave, feel free to drop off, but I will try to have y'all answer these questions. So question number 1 is, which HMDB ID version are you using for the harmonization?
Sure, I'll take this one. So right now it's using version 3, but it'll update to version 4 soon. So the primary difference there would be the coverage. They haven't changed, as far as I know, very many HMDB IDs, except that they have many more now than they used to. So we will update that, but we haven't done it yet.
Thank you, Steve. Question number 2. Does COMETS have metadata capture?
Evie?
Sure. So yes, I mean the metadata can be incorporated into your input file, and I would say that the more metadata you put, the better, especially for for the metabolites, because this is certainly going to help with harmonization, especially if you have HMDB IDs or PubChem IDs or other types of IDs. So yes, this information is captured, and it's also because it's included in your input file, and it's also output in your results.
Thank you, Evie. The next question: for data harmonization, do you start at the metabolite level, identified from a given study, or from the instrument level, example, metabolite peak level, etc.?
Ella, you got this one. I'll take the next one.
So the harmonization is done by Nathan from IMS, and he has an algorithm that looks at HMD first. So there's a hierarchy of I guess evidence that he uses to attribute. So he starts with HMDB ID, which is if you look at the UID01, which is the metabolite variable that identifies a given metabolite across COMETS, a lot of them start with HMDB something, and that's the primary, I think, the first level that he uses, and it goes from there to chemical ID, and I'm not sure if he's using retention time and mass charge yet. But we'll be documenting that very carefully, as we think that's also very important, and we can share that with you once we have finalized the documentation.
Thank you.
Could I
yes,
add
please.
to this real quick?
Please.
And so, it's mainly done at the metabolite level, but for the unidentified metabolites, depending on the platform, some of those may end up being metabolite peaks. And so those can certainly be analyzed, because remember that the software will run no matter what the harmonization status is. So it's still possible to look at them, but then of course in terms of the meta-analysis, they would probably be dropped out.
Thank you. The next question is, do you use a universal chemical identifier to map the metabolites across platforms? If so, what do you use? Is the harmonized metabolite database publicly available?
All right, so I'll take this one. So there's approximately 15 different labs that are contributing to COMETS cohorts, and we're primarily selecting, or actually at this point only selecting, cohorts with identified metabolite data. By identified, of course, different labs use different identifiers, which can make it a little bit challenging, or different names. So what we have done is collected from each of the labs kind of like a sample spreadsheet to see how much metadata they're including and how we can link across. And what we found was that each lab on average, in addition to including their own name and a standard chemical name, they will also usually include at least 2 to 3 unique chemical identifiers from online databases. They're not all the same across the different labs, but there's actually enough overlap so that if you have kind of all the files in hand, you can put it together like a puzzle. If A links to B and B links to C and so on, you can link all those unique identifiers to get one master list. Now, with that said, there's still a lot of what I call corner cases, things that are a little bit odd, like when someone calls glutamate L-glutamate and someone else calls it R-glutamate and someone else just calls it glutamate, right? And so typically we would maintain those as 3 separate metabolites, actually, because for the metabolite that's called glutamate, we don't know for sure whether it's L or R. But by and large, we've managed to pull together these links to come up with a relatively streamlined list. And we do have our own unique identifier that we then give each of those metabolites. And yes, it is publicly available. Actually, on that COMETS Analytics page, right underneath the button for download sample input was another button that says, hold on, it says download master metabolite list.
Yep.
So it's public right now. Anybody can just go on and grab it. That's our current version. By current, I mean the version that's in production. We actually have another one that we're about to include in the new production, probably in about a week and a half, 2 weeks time. So it's going to be a work in progress. We're going to be continually updating it. But yes, we do keep it public, and we welcome any comments.
Thanks, Steve. I think you just answered this, but I am going to ask it again just so it's clear to everybody, and then there was an additional question at the end of this. When will software be available to parties not in COMETS like the FDA, and can slides be shared?
Yes and yes.
Yeah, so it's available now. All you have to do is register yourself. You know, each person can have their own registration, and it's publicly available, comets-analytics.org. You just need a registration.
And for the slides, how would you like us to send those out? Just if anybody's interested in them, they can just let me know. How would you like to do that, the speakers?
That's fine.
That's fine.
Okay, so just let me know. I think most of you know how to get a hold of me. This is Krista, sorry. I didn't ever identify myself. This is Krista. If you do want a copy of them, just let Elisa or I know, and we'll get those to you. Okay, I did get 2 more questions, so I am going to ask those. If people do need to drop off, feel free to drop off. We'll only take these last 2, but I do want to give people the opportunity to get their questions answered. So the next question is, are QC samples like pooled samples or NIST standards used and analyzed in determining quality of data?
I'll take this one. So not currently. What we have done is send some samples to both labs in common, so split samples, and compare the reliability of metabolite measures from lab A to lab B. And we have had generally good results comparing, for instance, the Broad Institute versus Metabolon. The median correlation for that analysis over about 130 metabolites in common, the median correlation was 0.8. So we feel very reassured about that, but let me add that we do plan to include QC samples for future projects. So in a consortium like this where we're analyzing, previously collected data by and large, you can't just go back and add QC samples. It doesn't work that way. But for projects going forward, we believe that we will be able to do that, which would allow us to compare measurements between platforms, at least to some degree. So the plan is to include about, I think it's 30 to 40, and Krista probably knows better than I do, but 30 to 40 samples from different individuals in large data sets. So if a study of 1,500 samples is going out, you put these 30 samples in, you send the data from those 30 samples to a central repository, and as other studies run and they send more data to that central repository, you can start to compare metabolite levels across different platforms and different studies. That's the plan.
Yes, thanks, Steve. And it is going to be, right now we have 30 samples in-house. There will ultimately be 40 samples. So Steve's numbers were right on. And then the final question, how secure is it to send data to the Amazon server for analysis? What type of encryption and other security measures are used?
Not it.
Yeah.
So as you noticed, the URL is actually HTTPS, so that means it's an encrypted connection to the Amazon Web Services. And this group from CBIT, which is, super professional, and have set up the servers to be very secure, in their environment. I think it's pretty secure, but again, there's always you have to take that with a grain of salt, but we tried to make it as secure as we could possibly can. And, you know, data is not stored there. You know, it just runs through the algorithms that we have put together in COMETS Analytics and, you know, releases that data. Nothing is stored except for the files that Steve talked about.
And just to add one thing to that, we do request that the cohorts use de-identified data. That's standard practice for epidemiologic studies.
Thank you. So I'm going to thank you all for your attention and active participation in today's webinar. We would like to take this opportunity to thank our presenters, Dr. Evie Matei, Stephen Moore, and Ella Temprosa, for an excellent presentation. And we welcome your feedback to inform future webinars. So again, thank you all very much for joining us today. Thank you to our speakers, and everybody enjoy the rest of your day.
Thank you. Bye-bye.
Bye, everybody.